Now that we've talked about nitrogen sources and additions, it's time to shift focus to nitrogen transformations. And transformations are literally as they sound, again, thinking about this box that we are assuming is our soil system. Transformations are forms of nitrogen that essentially change form or change the state of existence in the soil itself, okay? And so in our pictorial nitrogen cycle, we're going to really talk about just a few of these: mineralization, nitrification, immobilization, and ammonium fixation. Okay. So again, here they are: the mineralization, mobilization, fixation, nitrification. Okay, and we'll start with mineralization. So, and mineralization again is the conversion of an organic form of nitrogen to ammonium. We can think of mineralization essentially as the process of decomposition. It's turning organic N, which again is the vast majority of the soil. Soil organic matter is organic N, and it's driving that into an inorganic form. Okay, and so there's really two steps here. There's this aminization, converting proteins to amino acids, amines, and urea. So it's taking proteins and peptides and breaking them up into smaller subunits. And then the second step is ammonification, i.e. the production of ammonium, okay? And this further converts organic N compounds to ammonium itself. Once it's in that ammonium form, again, it's subject to several potential fates, to nitrification, to plant uptake, immobilization, and fixation or volatilization. So once we have ammonium, there's a lot of different potential fates that can happen, and that's one of the reasons. One of the reasons that nitrogen is very tricky to manage, because of the multiple phases that it can exist, and the multiple pathways for loss or even just for transformation that occur. Mineralization is affected by things that you would think affect soil biology and microbial activity, and this is true of really all of life. Life is affected by temperature, soil pH, and water concentration, and soil microorganisms are no different, right? And so again, we can think about kind of chemically this two steps of aminization and ammonification. Aminization is taking a protein and then breaking it down into things like amino acids, to amine groups, or even into urea. So these are potential byproducts or outcomes of this aminization step. And then ammonification, taking amine groups, amino acids essentially, and with water we're essentially generating NH3 ammonia. That reacts very quickly with ammonium with it's a hydrolysis, splitting of water, and it's making this NH4 and OH. So this is the reaction of actual ammonium production. It's very common in soil. Mineralization is driven by a number of things, but water and temperature are really two primary pieces to that. Here we've got on the vertical axis nitrogen mineralized in milligram per kilogram or part per million. And then we've either got incubation time as the x-axis, and then we either have water holding capacity or temperature. And so we can see as the water holding capacity increases to 100, 50, 70, 90%, 100% would be essentially field capacity. In other words, when water is holding as much, soil is holding as much water as it can without gravity pulling it out. And then as temperatures increase from 10 to 15, 15 to 20, and 20 to 25, our rate of mineralization essentially increases, okay? So as water increases and as temperature increases, mineralization processes increase, and that should be no surprise, all within reason. Of course, if we really drove the temperature up and really made the soil saturated, this trend would reverse. It would plateau out essentially. So, okay. Just this concept of what total nitrogen is, or what soil nitrogen is. There's a very good relationship between organic matter and total nitrogen in soil. Organic matter is not just nitrogen, of course. Maybe 50% of organic matter is carbon, but as organic matter increases, by default, our total nitrogen increases too. Okay, so when we have more organic matter, we have more nitrogen, and when we have more nitrogen in the soil, typically we're going to have more nitrogen mineralized. These are relationships that are probably from the textbook, probably from similar soils. In the real world, if we went out and measured this on many different soil types, the data would look messier, but it's just illustrating the point that as our organic matter increases, our total nitrogen increases, and then as our total nitrogen increases, organic nitrogen mineralization increases. So in this idea that when we have more organic matter in the soil, we're typically going to have greater N mineralization. When we think about mineralization, it's important to think about kind of seasonal trends, especially in our environment in Ohio, this temperate environment where it's punctuated by nice warm summers and then frozen winters, and then the spring and the fall in between. So when we are talking about nitrogen synchrony, nutrient synchrony is an important concept, right? Thinking about N management. And what do I mean by that is that actually matching up the supply of nitrogen from the soil with actual plant demand, okay? So here we have a general relationship, this dashed line. As we come out of winter and into early spring, and then into late spring as soil temperatures really warm up, our nitrogen mineralization from that soil, the soil organic matter starts to decompose and mineralize. Our rates of N mineralization really increase, okay? And that's what's showed by this bell-shaped curve here. We have winter wheat and corn as different nitrogen uptake patterns by those crops, okay? So, of course, wheat being a cool season grass, it grows quite rapidly in early spring, and corn being a warm season grass, it's going to grow the fastest with highest N uptake rates in, say, July and August. And ideally, we could grab these crops and align them perfectly with actual what's happening in the soil for N release, but that's unfortunately not the case, right? So one of the challenges, or one of the things, and there's a lot of work that's done on this, thinking about cover crops and termination and nitrogen release, is can we match up when the soil is going to be releasing nitrogen, releasing that inorganic nitrogen, and when the plant's going to need it. So, we need to just recognize that these things aren't always matched up, and oftentimes they're not. And then just, also recognizing that nitrogen uptake for crops can vary quite a bit, right? So we saw it here, where there's the first, say, month there's not a lot of uptake, and then it goes into this really large, very large uptake period, right? Where there's a rapid nitrogen uptake. And this is exactly kind of what this graph is showing. So it's percent, this is all based on a percentage of the total. And we can actually look at the rate. So as we get into this kind of V10, V8 period, vegetative 8 or 10, our nitrogen uptake rate, this blue line indicates uptake rate. It gets up to, maybe 3-4 pounds of nitrogen per acre per day. It's just a tremendous demand that corn crop is placing on the soil and taking up as much nitrogen as it can, right? And this is one of the reasons, not surprisingly, that corn is a very nitrogen demanding crop, and most people are aware of that. And it's interesting, this is just a nice graph, kind of while we're still on corn, to think about what the plant is actually doing physiologically. So it's not just growing kind of amorphously. The plant has strategy, and this is, again, nitrogen uptake over the course of a growing season. We can see after, say, V8, V10, it really gets into this kind of exponential growth phase. It's pulling a lot of nitrogen up, but it's not until really tasseling that we see any ears appear and any grain appear. So what the plant is doing, it's storing a lot of N in leaves, in sheaths, in stalks, etc. And then as that ear forms, it's translocating, physically taking nitrogen, proteins, amino acids, translocating those, oftentimes breaking those down into smaller subunits from these storage molecules and shuttling it or shunting it into the grain itself. And this is true for really lots of grain crops that we deal with. And it's true for, a lot of plants that do a rapid vegetative growth phase. They kind of store as much as possible, and then they'll put that energy, that reserve, and those nutrients into its reproductive parts. Okay, so vegetables do kind of similar things, maybe not to the same degree, and it depends on the crop, etc. But we can see there's this huge reservoir in the leaves that occurs and in the stalks, and then this ultimately, not all of it, but a lot of it gets translocated again into this grain as that nitrogen fraction in the grain grows and grows until harvest. Need to recognize that plants respond to changes in soil available N. Okay, so this is some nice data from, a little old but still highly relevant, looking at alfalfa in a first, second, or third cutting. Okay, and you can see what I've just highlighted here: N, nitrogen from fixation, from biological fixation, and nitrogen from the soil. And this was quantified over the growing season of the first harvest, second harvest, and third harvest. So maybe a late spring, early summer harvest, a midsummer harvest, and then, say, an early fall harvest. And we can see the percentage from nitrogen fixation really changes over the course of that growing season. So in the first cutting, about 50% of the nitrogen is from fixation, okay? And why is that? Well, it's because 50% or 54 pounds per acre are coming from the soil. That's when soil is mineralizing in that, say, May, June period, that late spring period when there's a lot of available soil nitrogen. So this alfalfa crop does not need to fix a lot. It doesn't need to invest a lot of energy in photosynthate for the rhizobia to fix a lot of nitrogen. But as that mineralization curve kind of dries up and soil nitrogen becomes less and less available, because soils typically dry out, a lot of that nitrogen is either lost or it gets taken up by plants or immobilized into microbial tissue. So it goes from 54 pounds per acre to 22 from the soil. And that is met with a much larger fraction coming of nitrogen needs of the plant being met by fixation. Okay, and so then again in the fall we see an actual drop in that relative percentage because typically the soil moisture is going to increase at that, and we can look at this total yields. First cutting with the highest yield, second cutting. And then this last cutting is a really low, so it's less than half of that total. So there just wasn't a lot of growth in this scenario. Suffice it to say that plants, not just legumes, but plants in general, are going to respond to changes in soil nitrogen. Okay, so immobilization, again, this embodiment of nitrogen into tissue. By the textbook definition, it's the conversion of inorganic N, that is ammonium and nitrate, into an organic fraction. Microbes eat first, and this is like a really important concept when we think about managing nitrogen, is that microbes have very porous membranes. They have very high, you know, you can think of a microbe essentially as a sac that's built, when it wants to eat, it basically excretes enzymes extracellularly outside its cell, and then it sucks in what it needs, right? And this is one of the magics of soil and why microbes are really important. But microbes, in lots of ways, are very competitive with capturing any available organic nitrogen. So in this hypothetical scenario, if you've got a crop that you're trying to fertilize that needs nitrogen and microbes that are there and need nitrogen, the microbes are going to eat first. And that's really kind of goes into this idea of microbes are more capable of immobilizing N quicker than plants are. Okay. And when we talk about immobilization, it's really important to consider carbon to nitrogen ratios. And this is something that, is talked a lot in ecology. It's talked a lot about in biogeochemistry. This idea of stoichiometry, or the ratios of particular nutrients in a system. Microbes themselves, bacteria, have a carbon-nitrogen ratio of maybe somewhere between 8:1 and 12:1. That is, every carbon, for 8 carbons there's 1 nitrogen, or for 12 carbons there's 1 nitrogen. Plant residue, when we add that to soil, if it has a low nitrogen ratio, C to N ratio, it typically means there's more nitrogen for every carbon. So this is can be counterintuitive if this is the first time you've experienced or seen this, but low C to N ratios typically mean that material is more nitrogen rich. It's like a better quality material. If you have a high C to N ratio, you have low quality material. But this idea of 20 carbons to 1 nitrogen is this quote-unquote magical cutoff number. It's not really that magical in the real world, but it's a good rule of thumb that if it's less than 20 to 1, it's going to mineralize and release nitrogen, and if it's greater than 20 to 1, it's going to lock up and immobilize nitrogen. So this is important because we think about, if we're going to think about, and I guess we can talk about this in a second, but for adding amendments or something, compost or some sort of organic material, a cover crop, etc., to a field, and we want to be careful about it not robbing or immobilizing nitrogen from our cash crop, then we have to think about what the C to N ratio of that material is, and is it going to be a net immobilizer of nitrogen or a net mineralizer of nitrogen? So here are just some selected C to N ratios. We can see things like clovers, young clovers 12 to 1, clover residues 23 to 1. We get into like grain straw or grasses 60 to 80 to 1, and then going all the way up to like oak trees, sawdust 400 to 1, and then spruce and fir, which are just really mostly just carbon, oxygen, and hydrogen. That's all they really are, and a teeny bit of nitrogen peppered in. But for every, for fir here, it's suggesting 1 nitrogen for every 1,200 carbon atoms, which is, quite low. So carbon-nitrogen ratios, there's a lot of things that drive mineralization and immobilization dynamics, but one of the best indicators is this idea of carbon to nitrogen ratios. And so here on this vertical axis, this y-axis, we've got immobilization or mineralization, 0 being no net mobilization or mineralization, plus being mineralization, and minus being immobilization. Oh, wait a second. I think I just said that wrong. So, so this is nitrogen content, of the residue. As that nitrogen content increases, we're going to have more nitrogen actually mineralized. So this positive is a mineralization and negative. I think I just said that wrong again. Okay, so what we're talking about here is when the nitrogen content, this is a concentration. So as the concentration increases greater than this 20 grams per kilogram, that is essentially 2%. When it's greater than 2%, we have positive values or mineralization. When it's less than 2%, we have negative values or immobilization. Sorry about that, folks. That's a little confusing. I got mixed up on that. So I also wanted to show you, this an illustration. This is an interesting experiment when they basically took different residues. They buried it in a soil, and then they in a pot experiment, and then they measured how much nitrogen was taken up by the plant. Okay, and so here's the different C to N ratios of various plant residues, and then how much nitrogen is coming off in the measured plant. So things like collard roots, bean stems, tomato leaves, beans, with these respective C to N ratios, we're able to release nitrogen, mineralize nitrogen, and plants took those up. So going from anywhere from 2.2 milligrams to 0.3 milligrams. But then when we drop below this 20 to 1 ratio, or above the 21 to ratio, so when the C to N is 27 to 45, you can see that there's essentially an order of magnitude drop here. It goes from 0.3 to 0.03 and less in terms of total nitrogen uptake. So, this idea, it's a nice rule of thumb. It's, of course, not perfect, 20 to 1 for carbon-nitrogen ratio, but it is a helpful number to have in mind. All right, now let's think about ammonium fixation. We mentioned this a little bit earlier, but this is when 2 to 1 clays are capable of fixing ammonium by replacement. So this goes back to some of the concepts we talked about in the first week or second week with clay lattices and clay layers and being able to trap cations in there. Now ammonium can be held superficially on exchange sites, and it can also be held more kind of intricately or intimately in these clay layers. Okay, so things like vermiculite and micas are responsible or behave in this way. Not all clays are clays vary in their ability to fix ammonium, but 2 to 1 clay minerals are much better at it, or much more likely to behave this way than 1 to 1 clay minerals. And so ammonium can come in and knock out things like potassium or calcium or magnesium, these cations, and they can get replaced. So, this is typically, we don't think that this is a huge net kind of fixation phenomenon in soils. A lot of our managed soils, there might be 1, 2, 5% of our total ammonium that's fixed, but it's still a potential pathway for transformation. And it really, you can think of it kind of like storing it in a sponge of sorts, and then ultimately that's going to be released back into the system, but it might not be till next week. It might be next month, next year, next decade, et cetera, et cetera. All right, and then nitrification. Again, this is another jargon word, right? But it's, if we think about nitrification, just as the production of nitrate. So this is a major product of what happens after ammonia and ammonium are produced through mineralization. It's this conversion of ammonium to nitrate, and you can see this step with nitrite, NO2-. Again, the idea of why do microbes do this, they're basically oxidizing ammonium to get energy out of the chemical bonds between the nitrogen and these 4 hydrogens. So it's a biologically driven process, and it's affected by a number of things. Well, first of all, not surprisingly, the concentration of ammonium in the soil. the actual critters that are there, the nitrifying organisms, what the pH is. Higher pHs tends to be driven. Soil needs oxygen for this reaction, and so if it's an anaerobic soil, you're not going to have any nitrification. But if it's an aerobic soil, well aerated soil, you will have it. Soil moisture, again, for the biology, and then soil temps. These are all things that will drive nitrification or help influence it. Two steps driven by two unique sets of bacteria, Nitrosomonas in the first step in the production of nitrite, and then Nitrobacter in the production of nitrate. Okay, so different critters and different enzymology. Nitrification is related to soil pH. As soil pH increases, nitrification rates and percentage of total nitrification are going to increase. And we already mentioned as moisture increases, cumulative nitrate production increases as well. That's exactly what this graph is showing. So as pH increases, as moisture increases up to field capacity, nitrification rate of nitrate production will increase. Okay, so after nitrification, then what? Well, nitrate, again, is this anion, and it's has a number of potential fates. It can go into soil solution and be taken up by plants. It can be lost through gaseous form via denitrification, and it can be lost through leaching. So again, these are, we're going to wrap up here with the kind of transformation piece, talking about mineralization, nitrification, immobilization, and ammonium fixation. So those are those four pieces represented as 2, 3, 5, and 7 here in this nitrogen cycle diagram.